Hello everyone. In this video I'm going to explain for you about meiosis, which is the division of the gametes or the sex cells in the body. So first of all, it's good to remember that meiosis is the second type of cell division in the body. Recalling that we have mitosis, which is the division of the body cells, and meiosis, the division that result in the production of the sex cells or the gametes. So what is meiosis? And how it's different from mitosis, and what are the steps of meiosis, and this is what we are going to discuss in this video. First of all, it's very important to know that at some point during the division of the cells must maintain their chromosome number. As a result, mitosis will make sure that all the body cells have the same number of chromosomes, which is 46. But if this is applied to the sex cells, the sperm and the egg, then during fertilization, the sperm cell and the egg cell, each one carrying 46 chromosomes, will result in an embryo with 92 chromosomes. And speaking about the number of chromosomes, if the number of chromosomes changes, it means we are speaking about another species, because the number of chromosomes is considered a constant characteristic for species. So humans have 46 chromosomes, chimpanzees have 48, dogs have 78, etc. So in order to make sure that the embryo or the zygote maintains the same number of chromosomes, which is 46, then we need to have 23 chromosomes carried by the sperm and 23 chromosomes carried by the egg. And for this reason, there is meiosis. So meiosis is a type of division that reduces the number of chromosomes from 46 into 23 chromosomes. So as we said, gametes or sex cells, they must have half the number of chromosomes that we have. In case of humans, we are speaking about 23 chromosomes. Speaking about type of cells, the body cells or the cells in the body are divided into two groups. We have the somatic cells and the gametes. The somatic cells are all the body cells except the sex cells, including the bone cells, the nerve cells, skin cells, the muscle cells, blood cells, white blood cells, etc. All these cells, they divide by mitosis and they have 46 chromosomes. On the other hand, in the ovaries and testes, which are the reproductive organs, we have something called germ cells. These cells will develop into gametes, or the reproductive cells or the sex cells, which are egg in female and sperm in male. These cells must have half the number of chromosomes, and they are the only cells that divide by meiosis. So in order to understand that, let's ask, how can we observe or how can we study chromosomes in the cell? Because we are speaking about the number of chromosomes in each cell. So there is a technique called karyotyping. Karyotyping allows us to see the chromosomes or observe the chromosomes in a certain stage in the life cycle. And by comparing the karyotype of a somatic cell and a gamete or sex cell, we can see the difference, that in the somatic cell we have 46 chromosomes, whereas in the sex cell we have only 23 chromosomes. So this is the karyotype. And usually karyotype, it's a diagram taken when the cell is dividing and when the chromosomes are visible during metaphase mainly. So going back to cell division, you remember that during mitosis the chromosomes are visible so that they can separate. So karyotype usually is taken from cell who are actively dividing, because if a cell is still during G0 or during interphase, the chromosomes won't be visible. Instead, the DNA will be as chromatin material. Okay? For this reason, this technique, karyotyping, is taken from cells that are actively dividing. So by looking at the karyotype, you can see here we have 1, 2, 3, 4, a total of 46 chromosomes. Okay, and they are labeled as numbers 1 to 22, and I have two chromosomes. Usually they are put at the lower corner of the image. Those are the sex chromosomes. Now, speaking about that, so what do I mean by sex chromosomes, and what are the rest? The rest are called the homologous pairs, okay, or the autosomes. Those are 22 pairs, and they are homologous. What do I mean by homologous pairs? So As your DNA or your chromosomes are half taken from your father and half taken from your mother, but we share the same chromosomes, we share the same genes, but different alleles or different information. So chromosome number 1 from the father and chromosome number 1 from the mother are the same, but they carry different alleles, they carry different DNA sequence, but they carry information for the same traits, for the same genes. For this reason, the karyotype will arrange your chromosomes as parental, paternal and maternal, one from your father, one from your mother, beside each other based on size. So if you look again at this image, you will see that chromosomes number 1 are the tallest among the chromosomes. Okay, so these chromosomes from 1 to 22 are common between male and female, and they carry characteristics that are common between males and females. For example, the function of muscles, vision, information related to digestion, to the function of the brain, information related to maybe tasting or hearing or production of certain enzymes or hormones, okay, that are common between males and females. So all the information carried on the autosomes are common between male and female. So if there is any mutation or any change or any mistake happening at the level of these autosomes or the 22 pairs, it's not going to be gender based. Okay, it can be common or it can be distributed equally between males and females. On the other hand, we have only 2 chromosomes, we call them the sex chromosomes. These chromosomes are related to gender, and actually these are the chromosomes that determine the gender of the embryo during fertilization. These chromosomes are X and Y, and males have XY, females have XX. Okay, so if we ask ourselves, males have Y chromosome, females don't have Y chromosomes, we can come to a conclusion that the Y chromosome contains the genes responsible for the primary and secondary characteristics of the males, including the formation of the male reproductive system, the appearance of the secondary male characteristics, including the beard, growing of the beard, muscles, hair in the body, etc. Okay, these characteristics are exclusive for males because they exist on the Y chromosome, and females don't have the Y chromosome. Okay, so males have XY, females have XX chromosomes, and this is important in order to determine the gender of the embryo. Going back to the types of cells, the diploid cells or the body cells are the cells that have 46 chromosomes. Even we refer to them as 2N cells. 2N, it means having 2 copies of the chromosomes, one from the mother, one from the father. Okay, and these cells, as we said, they are called diploid cells or the somatic cells. The other types of cells are called the haploid cells or 1N cells. They have 23 chromosomes in humans. If we are speaking about any other species, they will be having half the number of chromosomes of the body cells of that species. For example, a chimpanzee will have 24 chromosomes in the sperm and the egg because they have 48 in the body cells or the somatic cells. So what is meiosis? Meiosis is the formation of these haploid cells in the body, and mainly in the reproductive organ, the testes in the male and the ovaries in the female. So the body cells divide by mitosis, as we explained before, and mitosis maintain the number of chromosomes. Mitosis will result, one mitosis round will result in 2 cells, 2 daughter cells that are identical to the mother cell, having the same number of chromosomes, same information, there is no variation. As a result, if a muscle cell divides, it's going to produce 2 muscle cells that are exactly the same, carrying the same information, the same number of chromosomes. On the other hand, meiosis produces 4 cells from one round. Okay, 4 cells, not 2 cells, 4 cells. And these 4 cells are not identical. And we are going to study later why they are not identical, what causes the variation in these 4 cells. Actually, it's a genetic variation. It means the DNA carried in each cell will be different. And this is the reason why you are different from your siblings. Because each time spermatogenesis or production of sperm happens and production of egg happen, totally different cells are produced. Okay? Except for the identical twins, because they come from one egg and one sperm. So identical twins, they come from one zygote that's split into two embryos. In order to understand more meiosis, we have to know that meiosis is divided into two divisions. The first division is called reductional division, the second one is called equational division. And as we said, the main idea of meiosis is to reduce the number of chromosomes. And this will happen in the first round of division, which we call it meiosis 1, where the chromosome number will be divided into 23 in each cell. In the second division, these two new cells that have 23 chromosomes will undergo a division we call it equational, which is very similar to mitosis, that will split this cell into two cells. This cell into two cells having 4 cells. Now, later on, we will see the differences between this process, meiosis, in male and in female. There are some differences at the level of the number of chromosomes, at the level of the number of cells that are produced. Actually, in females, even though we are saying 4 cells are produced, but only 1 cell will be the egg, will take most of the cytoplasm, whereas at the other hand, in males, 4 sperm cells are produced. We will come into that in the coming slides. So what is the result of meiosis? 4 haploid cells with n number of chromosomes, and these chromosomes at the end, there will be single chromatids. Now, genetic variation is a very important event, or very important, let's say, very important outcome of meiosis. And it is the result of 2 events that happen during meiosis. So again, what is genetic variation? It means having cells with different DNA or genetic makeup. And this doesn't exist in mitosis, because mitosis produces cells that are identical, but it happens in meiosis. So what is the reason why during meiosis, what happens to result in cells that don't have the same DNA or have different variation of DNA? The main reason is the occurrence of 2 events that happen in the first set of division, which we called it reductional division. These 2 events are called crossing over and independent assortment. Crossing over and independent assortment are 2 let's say events that happen during meiosis 1 that will lead to genetic variation. Okay? So what is crossing over when it happens? What is independent assortment when it happens? This is an image of crossing over. As can you see, we are here showing homologous chromosomes. It means one from the father, one from the mother. And this is crossing over. If we look at this image, we can conclude what is it. It's exactly exchanging of fragments between the father and the mother, or the paternal and the maternal chromosomes in the cell. It means we have one chromatid which is going to be recombinant. What do I mean by recombinant? Yani carrying information from the father and from the mother at the same time with different percentages. So what it means is, if this recombinant chromatid is going to pass to the embryo is going to have from the same chromatid information from the father and from the mother. Okay? And this will cause genetic variation. And